Hello, my name is Claire, working with Miguel and Isabella in the Microbial Biotechnology Division of the Research and Design Program here at GCU under the direction of Dr. Kufrick. Our research entails the photosynthetic processes in cyanobacteria and their contribution to the bidirectional hydrogenase enzyme. We are interested in the biological production of molecular hydrogen to be used as a clean, non-carbon-based source of energy. Cyanobacteria are able to produce molecular hydrogen, and we can use that molecular hydrogen in a hydrogen fuel cell to generate energy, as with water being the output instead of greenhouse gases. Hydrogen is also of interest because it is the highest energy dense of all fuels, and it can serve as an alternative to many fossil fuels, making it a sustainable source of energy and therefore our interest.
In order to create more molecular hydrogen in order to be used for hydrogen fuel cells, we use a specific species of cyanobacterium called Syncystis SPPCC 6803, which essentially has this specific enzyme called the bidirectional hydrogenase enzyme. And what makes this species of cyanobacterium special compared to others is has both a forward and reverse reaction, but leaning towards forward reaction, essentially oxidizing to create molecular hydrogen. It does this by having different subunits such as HOX-H, which has a nickel iron core, which deals with oxidation and redox reactions, but we use photosynthetic reactions in order to drive a backwards reaction to create molecular hydrogen.
The current HOX promoter in our bidirectional hydrogenase does not have a high expression rate, resulting in a relatively low expression of our bidirectional hydrogenase in the cells. So in order to do this, we first clone the operon using PCR in the region containing our operon of the Syncystis genome. We use our PUG19 plasmid as a vessel for cloning and creating our HOX operon cloning construct, which will actually be cloned in E. coli. So when doing our PCR, the region that contains our operon is actually PSHA1 and AFI1, which will then be inserted into our PUG19 plasmid in the region of HINC2 and SFO1. This will overexpress our bidirectional hydrogenase and will increase the molecular hydrogen output. This is very important for our research, as we can harness the increased output to be cultivated in biofuel.